This second podcast on our series on the endocrine organs will deal with the pituitary gland and the hypothalamus. The pituitary and hypothalamus are structurally and functionally linked in the endocrine and neuroendocrine control of other endocrine glands. Now, the pituitary is also called the hypophysis. The pituitary gland plays a central role in regulatory feedback, and you'll see this in much greater detail in your physiology course. If we look at the pituitary gland, it's actually two organs in one with two distinctly different embryologic origins. The anterior lobe of the pituitary is called the adenohypophysis, and it's a typical glandular epithelium. The posterior lobe, also called the neurohypophysis, consists of neurosecretory tissue. The anterior lobe develops from the ectoderm of the oropharynx, so-called Rathke's pouch, the pars distalis, which makes up most of the organ. There's a pars intermedia, which is a remnant of the posterior wall of Rathke's pouch, and the pars tuberalis from the lateral wall of Rathke's pouch forms the sheath around the infundibulum. The posterior pituitary develops from the neuroectoderm of the floor of the third ventricle, the so-called diencephalon. The posterior pituitary consists of the pars nervosa, which contains neurosecretory axons and axonal endings, as well as glial cells, and then the infundibulum contains neurosecretory axons that originate in the hypothalamus. The pituitary is a relatively small organ, maybe about a half a gram. It's considerably larger in women that have had two or more children. It resides in the sella turcica. The infundibulum and a vascular network connect the pituitary to the hypothalamus, and we'll show that in a few minutes. The posterior pituitary, shown on this low-power histological view, then contains the pars nervosa and the infundibulum. The anterior pituitary contains pars intermedia here, the pars distalis, which is the majority of the lobe, and then the pars tuberalis. And you can see that on the diagrams, so the anterior lobe and the posterior lobe. And this would be diagrammatically the hypothalamus, and we'll talk about these nuclei in the hypothalamus later. And remember, from the point of view of the nervous system, when we talk about nuclei, we're talking about collections of nerve cell bodies. This is just an overview of the development of the pituitary. You would have discussed this earlier in your training, so it's not so important for our course. I just put it here to remind you of what you've already learned. But if you look at the development of the pituitary from Rathke's pouch and the neuroectoderm, if you're looking at this in a time sequence from A to B to C, this may be occurring between the third week of fetal development up until maybe the 8th week of fetal development. The blood supply to the pituitary, this is fairly important. Most of the anterior pituitary has no direct atrial blood supply. The superior hypophyseal arteries, which arise from the internal carotids and the posterior communicating artery of the circle of Willis, will supply the pars tuberalis, the median eminence, and the infundibulum. The inferior hypophyseal arteries, which also arise from the internal carotids, supply the pars nervosa. The hypothalamic hypophyseal portal system is a critical link between the hypothalamus and the anterior pituitary, and it's important that you appreciate and understand this blood supply as you begin to put together what you're going to learn in physiology as far as the control of secretion from the pituitary gland. The superior hypophyseal artery gives rise to a capillary bed that drains into the hypophyseal portal veins, and this gives rise to a secondary capillary bed in the pars distalis. This system of vessels is going to carry neuroendocrine secretions to cells of the pars distalis. The anterior lobe regulates other endocrine glands and some non-endocrine tissues. And we're going to come to appreciate that there are distinct cell types in the anterior pituitary. It turns out that there are 3 categories of cells and actually 5 functional cell types and the hormones. Now, the categories of cells, basophils, acidophils, and chromophobes, are actually archaic terminology today. They were defined earlier, maybe middle of the 20th century, when we did not have the ability to do cytochemical stains for the different hormones, and they were defined basically on staining characteristics. So we talk about basophils staining basophilic, acidophils staining acidophilic, and chromophobes having no stain at all. But with more sophisticated cytochemical techniques, one can now identify specific cell types. So there are gonadotrophs, thiotrophs, and corticotrophs that are clustered together as basophils. There are somatotrophs and lactotrophs that are grouped as acidophils. The gonadotrophs, about 10% of the parenchymal cells in the anterior pituitary, secrete FSH and LH. Thiotrophs, about 5% of the parenchymal cells, secrete thyroid-stimulating hormone, also known as thyrotrophin. And the corticotrophs, maybe 15 to 20% of the parenchymal cells, secrete adrenocorticotrophic hormone. They actually secrete a precursor known as pro-opiomelanocortin, or POMC. The acidophils, which make up the vast majority of the parenchymal cells in the anterior pituitary, secrete growth hormone or somatotrophin, and then lactotrophs, maybe 15 to 20% of the parenchymal cells, secrete prolactin. Now, the chromophobes are degranulated cells. Their cytoplasm does not stain, so they're thought to be actually spent cells, either of the 2 categories, acidophils or basophils. The anterior pituitary, the cells are arranged as typical endocrine secretory tissue. They're in clumps or cords. They're associated with fenestrated sinusoidal capillaries, and the cells in the anterior pituitary respond to signals from the hypothalamus. All the anterior lobe hormones turn out to be small proteins. The anterior lobe hormone secretion is under regulation from releasing hormones from the hypothalamus. We'll talk about that in a little bit, and you'll learn much more about that in your physiology course. Now, there's also a group of cells that are called folliculostellate cells in the anterior pituitary. These cells are difficult to identify on histological images. They don't secrete hormones, but it's thought they may play a role in the local release of anterior lobe hormones. Perhaps they release some cytokines, and by paracrine secretion may stimulate some of the other tropes in the anterior pituitary to release their hormones. This is just a typical histologic preparation that's stained with a variety of different stains to highlight acidophilic staining cells, basophilic staining cells, and cells whose cytoplasm doesn't take up any stain, the so-called chromophobes. Here's another histological preparation, stained here with a Mallory trichrome stain to clearly highlight the acidophils with the acidic granular cytoplasm, the basophils with the basophilic granular cytoplasm, and the chromophobes showing no stain at all. This is a nice image because you can clearly see the blood capillaries that are associated with these endocrine cells. This is a table that shows some of the cells, hormones, physiological action, and the releasing hormones that influence secretion from the anterior pituitary. It is important that you understand and can be able to identify the cells and the hormones on this table. So, for example, the basophils, a so-called gonadotrope, thyrotropes, and corticotropes. The acidophils are the somatotrophs and mammatrophs. The gonadotrophs, for example, synthesize and secrete follicle-stimulating hormone and luteinizing hormone. For our course, I don't care that you know the ultrastructure of these cells, but I do want you to understand the physiologic action. So, for example, FSH and LH. FSH stimulates ovarian follicles in the female. It stimulates spermatogenesis in the male. Both FSH and LH do this. These hormones are under the influence of the hypothalamus. Gonadotrophin-releasing hormone stimulates their release from the anterior pituitary. Thyrotropes release thyrotropin, so-called TSH. This stimulates the thyroid gland. Thyrotropin-releasing hormone from the hypothalamus regulates its secretion. And you can go through this table so you can see ACTH for the corticotrophs, somatotrophin for the acidophils, prolactin for the mammatrophs. ACTH under the influence of corticotrophin-releasing hormone, somatotrophin under the influence of somatotrophin-releasing hormone, prolactin under the influence of prolactin-releasing hormone. So I want you to spend a little bit of time going over this table so that you can appreciate what the different hormones do. This will be important as you try to integrate what you learn in this course with physiology. Let's talk a little bit about the pars intermedia. And the pars intermedia, as far as function in humans, it's not well understood. The pars intermedia consists of small cystic cavities that are residual from the lumen of Rathke's pouch. These colloid-filled follicles are somewhat reminiscent of the thyroid gland. The cells in the pars intermedia may include basophils and chromophobes. In other species, some of these cells are thought to contain endorphins. In frogs, the basophils produce melanocyte-stimulating hormone. Melanocyte-stimulating hormone is not thought to be a functional hormone in humans. It is a byproduct of POMC, so it's thought that some of the cells that are in the pars intermedia in humans may be corticotrophs. You don't have to really worry about this particular stain. It's a toluidine blue stain. The take-home message from the point of view of humans is the pars intermedia. We don't really understand its function. The posterior lobe is an extension of the central nervous system, and it stores and releases secretory products from cell bodies that are in the hypothalamus. The posterior lobe, also called the neurohypophysis, is not an endocrine gland in that respect. We talk about the pars nervosa and the infundibulum as part of the posterior lobe. Now, the hypothalamic hypophyseal tract contains non-myelinated axons and endings that come mainly from the supraoptic and paraventricular nuclei that are in the hypothalamus. And remember, when we talk about these nuclei, we're talking about clusters of cell bodies of neurons in a brain region. Now, these axons don't synapse with other neurons or target cells. Rather, they release secretory vesicles containing peptide hormones, the so-called neurosecretory products. And these products are oxytocin and ADH or vasopressin. Oxytocin stimulates lactation by causing contraction of the myoepithelial cells in the lactiferous glands of the breast. Oxytocin also stimulates smooth muscle contraction in the uterus during parturition. As you know, ADH stimulates water reabsorption in the kidney. These prohormones contain peptide hormones plus neurophysin carriers, and these accumulate in dilated portions of the axons that are called Herring bodies, and I'll show you some images of that in just a minute. Now, it turns out that the pituocytes are really the only cell that's specific to the posterior lobe of the pituitary. These pituocytes are actually glial cells. So this is a diagrammatic view of an axon in the posterior lobe showing the so-called Herring bodies that are accumulations of these neurosecretory vesicles. Here's an electron micrographic view of the Herring bodies. You can see the neurosecretory granules. Here at a lower power electron micrograph, you can see what the Herring bodies would look like. In a histological view, this would be the posterior pituitary. Here are some of the herring bodies. This is a PAS stain. I don't care that you remember the stain, but you can see the herring bodies. Here, an aniline blue stain. You can see the herring bodies. Now, I want to caution you, on our glass slide collections, the herring bodies are exquisitely difficult to identify. So you just have to understand that they are accumulations of these neurosecretory products with the neurophycins, the so-called carrier proteins for the small peptide hormone products. The cell nuclei that you see in the posterior pituitary would be cell nuclei of pituocytes. These are essentially glial cells or supporting cells for the axons that are coming down from the hypothalamus in this region of the pituitary. And you can clearly see that there are blood capillaries associated with this region of the gland as well. Here's just a low-power histological view labeling the components of the pituitary. So the anterior pituitary like this, the posterior pituitary like this. So this is pars distalis. Here you can see a little bit of the pars intermedia with some of these colloid-filled follicles. The pars tuberalis like this, the pars nervosa, and the infundibulum in the posterior pituitary. A typical H&E type stain, one like you might see in our laboratory preparation of the anterior pituitary. You can clearly see the acidophils staining eosinophilic, the basophils staining more basophilic, and the chromophobes where you just see the nuclei staining but the cytoplasm not picking up any stain. So here's another example of some of the chromophobes. And here, the posterior pituitary looks particularly like nervous tissue. The blow-up of in the box, you can see pituocytes like so. These nuclei would be pituocytes. And here, probably a herring body. Now, as we said, the hypothalamus regulates the activity of the pituitary gland. The hypothalamus functions as the controlling center of the autonomic nervous system and the endocrine system. So the hypothalamus can regulate blood pressure, body temperature, fluid and electrolyte balance, and body weight and appetite. The hypothalamus is involved in neurosecretion by the hypothalamic hypophyseal portal system, so releasing hormones that influence the secretion of the anterior pituitary gland. These are polypeptides. These releasing hormones travel down the hypothalamic hypophyseal portal system. There's also neurosecretion of polypeptides from cell bodies and the neurons of the supraoptic and paraventricular nuclei that are involved in the storage of oxytocin and vasopressin from nerve endings in the posterior pituitary. Here is that table. These are the hypothalamic regulating hormones. So growth hormone, releasing hormone is actually, there are two forms in humans. There's a 40 and a 44 amino acid polypeptide. I don't care that you really remember that, but it's just for the sake of the discussion. The growth hormone, releasing hormone comes from the arcuate nucleus in the hypothalamus, and as you would expect, regulates secretion of growth hormone by somatotrophs. Somatostatin, two forms in humans, again 14 amino acid polypeptide, 28 amino acid polypeptide. Again, not so important that you remember that. Somatostatin comes from the paraventricular, periventricular, and arcuate nuclei in the hypothalamus. Somatostatin inhibits secretion of growth hormone by somatotrophs, and it inhibits insulin release by pancreatic islet cells. Dopamine from the arcuate nucleus inhibits secretion of prolactin by lactotrophs. Corticotrophin releasing hormone from the arcuate, periventricular, and medial paraventricular nuclei in the hypothalamus stimulates secretion of adrenocorticotrophic hormone by corticotrophs, stimulates gene expression for pro-opiomelanocortin in corticotrophs. Gonadotrophin-releasing hormone from these regions in the hypothalamus, arcuate, ventromedial, dorsal, and paraventricular nuclei stimulates secretion of LH and FSH by the gonadotrophs. Thyrotropin-releasing hormone, a very small polypeptide, stimulates gene expression and secretion of TSH by thyrotrophes, also stimulates synthesis and secretion of prolactin. This is just a diagrammatic figure to help you visualize the hypothalamic releasing hormones, their effects on the anterior lobe of the pituitary. So, for example, prolactin releasing hormone causing release of prolactin, prolactin affecting breast tissue, dopamine inhibiting the release of prolactin, and you can go through each of these. Now, one that I didn't mention, I talked about growth hormone releasing protein that stimulates the release of growth hormone from the anterior pituitary. There's also a growth hormone inhibiting protein. Now, feedback systems are important in regulating the endocrine function at two levels. You're going to learn about this in much more detail in your physiology course. Here I just show production of thyroid hormone as an example, and it's regulated at the level of hormone production in the pituitary gland and by the hypothalamic releasing hormones that are a product of the hypothalamus. So you can say that thyroid hormone is regulated through negative feedback. Thyroid hormone itself can feed back on the system and inhibit further release of itself, and this inhibition occurs at the level of the anterior pituitary and at the hypothalamus. Now, the system can be activated if thyroid hormone is low or if metabolic demand is suddenly increased. So there's both a negative and a positive feedback for these hormones from the anterior pituitary. Now, information from most physiologic and psychologic stimuli that reach the brain probably also reach the hypothalamus. So this hypothalamic hypophyseal feedback loop can provide a pathway for the central nervous system to regulate the pituitary gland and consequently the entire endocrine system. And this is also a primary mechanism by which changes in emotional state might be translated into physiologic or homeostatic states.